Hello, students. Welcome back to Chapter 9. So we started to talk about why our chromosomes are really the kind of foundation for our inheritance before I started having weird technical issues. So I'm going to pick up with that chromosomal basis of inheritance, and then also talk about sex chromosomes and genes that are linked to your sex chromosomes. So when we look at Mendels Laws, they are really kind of predicated upon the behavior of our chromosomes during meiosis, right? So genes occupy a very specific location, a very specific locus or position on the chromosomes. The chromosomes then sort of randomly arrange themselves and then sort of randomly segregate and separate. And Mendel's two laws, they really do correlate with this kind of chromosomal behavior. So for instance, when the homologous chromosome pairs sort of line up along the metaphase plate during metaphase 1, it's sort of very randomly, right? That leads us to the law of independent assortment in terms of Mendel's laws. And then when the homologous chromosomes separate during anaphase 1 in the first part of meiosis, that is the law of segregation. And so it is our chromosome behavior that really leads to how traits are inherited. So for instance, if we were to look at seeds for a pea plant, right? They're yellow and round. That's two different characters, two different traits. Well, here are the genes for those different characters, those different traits for those characters. And you have two equally probable arrangements of the chromosomes along that metaphase plate. You could have, red, red, blue, blue, or blue, red, red, blue. Like, it really doesn't matter. They're both equally possible. And then when the chromosomes separate during anaphase, they get pulled apart based on how they were arranged. And then when they separate again during meiosis 2, we end up with this sort of typical sort of ratio that we see where 9 times out of 16, they'll be yellow and round. 3 times out of 16, they'll be green and round. 3 times out of 16, they'll be yellow but wrinkled. And then they have a 1 in 16 chance of being green and wrinkled. That both sort of homozygous recessive. And it has to deal with sort of how these chromosomes sort of separate independently of each other. So one of the ways that scientists sort of look at chromosomal inheritance is by looking for things that sort of don't fit the pattern. And so two scientists, Bateson and Punnett, they studied plants that did not show that typical sort of 9:3:3:1 ratio in the F2 generation. They got a different sort of ratio of outcomes. And so what they discovered was that there are genes that are linked, meaning that these are genes that are generally located close together on the same chromosome and generally tend to be inherited together rather than sort of sorting independently. So for example, here we have a purple flower with long pollen. And they ended up with very, very, very different ratios. This was sort of the predicted ratio, but you can see they had way more offspring observed that had purple long seeds than, and significantly less of the purple round and the red long, and significantly more than the red round, sort of that the one that you would expect would be the sort of least abundant, the homozygous recessive. And so what they found was that these genes for the purple flower and the long pollen, the gene for flower color and pollen length, are located similarly on the same chromosomes and are inherited together. And so that leads to sort of that other sort of ratio that we get, and explains how sometimes genes are inherited together and not sort of independently assorting. Sometimes you can, right, part of our meiosis process is that crossing over where homologous chromosomes sort of switch genes and they switch genetic information. And when that happens, we can get new combinations of alleles in our gametes, in our sex cells. And so when we link our when genes get linked like this, they might get separated during a crossover process, and now they're no longer linked. And so sometimes what we typically look at is what is known as a recombination frequency. And so what is the percentage of the offspring that have sort of these recombinant sort of new combinations amongst the whole total. So here's our tetrad. There's our crossing over where they're switching the genes. So you can see here are the parental sex cells, but here are the recombinant sex cells. So this was sort of what resulted from the crossing over. So here, sort of the wild type, they did an experiment with flies. And sort of the kind of quote unquote wild type is a gray body with these very long wings, but sort of this recombinant there are also the black body with sort of minimal wings. And so these are our parents, right? This sort of wild type and this wild type female and this sort of wild type male. When they mate, we get sort of this resultant not offspring that in some instances resemble the appearance of the parents, but in some have these sort of recombinations where you have a gray body with small wings or a black body with long wings. And to determine the recombination frequency, you just add up the total number of recombinant offspring via the total number of offspring, and you get a percentage. And the reason we're getting these sort of recombinated DNA, the recombination, this new sort of version, is the genes get crossed over. Where some of the parental DNA gets put into the, gets switched over to the female, the maternal chromosome, and some of the maternal DNA ends up in the parental chromosome, sort of during that crossing over period. And so you end up with this sort of recombination and new traits appearing. When we are trying to kind of map our genes and sort of understand how genes kind of are located along the chromosome, which is what a genetic map is, very often we'll use these recombination frequencies. It sort of helps us determine the positions of different genes on different chromosomes. And when we do such a thing, the resultant is called a linkage map. And so you can see here is a section of our chromosome. It's carrying sort of these linked genes that will be inherited together. But by determining sort of the recombination frequencies, it sort of helps us kind of locate the genes and where they are located on a particular chromosome. And so you can sort of see that's what they've done here. Here are the sort of kind of wild phenotypes. And then here are sort of these resultant sort of recombinant types of offspring that have been made, because these are traits that would normally be kind of inherited together, but due to the crossing over are resulting in sort of different combinations and different appearances for flies like these. So the other sort of end of this chapter is about sex chromosomes and the genes that are linked to our sex chromosomes. So in mammals, the male has an X chromosome and a Y chromosome. The female gets 2 X chromosomes. The Y chromosome has all of the genes for the development of the testes. So the testes, testosterone, all of the sort of kind of male sex features, all of the genes for that are only located on the Y chromosome. And when a female inherits her 2 X chromosomes, since she does not have a Y chromosome, the ovaries are sort of allowed to develop then. Now, in mammals, it's all dependent on your chromosomes. And of course, there are people who have, XXY chromosomes and people who have, XX chromosomes. Male can get sort of 2 X chromosomes, so on and so forth. That's how it works in mammals. In sort of non-mammalian species, sex is not always determined by these sex chromosomes, but actually by temperature. So for instance, reptiles, sea turtles, for instance, that depends on the temperature at which the egg is incubated during a very specific sort of period of embryonic development, and that will determine whether there is a whole clutch or nest of male reptiles, sea turtles, for instance, or female turtles, for instance. And as the global temperature is changing, it is affecting sort of the ratio of males to females in many of these reptile species, which is then affecting the reproduction and sustainability of these different species. Because if you have many more females than males, or many more males than females, the species cannot sort of reproduce properly. So here you can see, here is our kind of sex chromosomes. You can see here is the male XY, the female XX. Her egg is only going to carry an X chromosome because that's all she's got. Whereas the male, when his gametes are formed, depending on how they split, his sperm might have be carrying an X chromosome, his sperm might be carrying a Y chromosome. If the sperm mates with an egg and they were both carrying 2 X chromosomes, then the resultant offspring will be female. If the sperm carrying the Y chromosome mates with and fertilizes the egg bearing the X chromosome, then you would end up XY again, and the resultant offspring would be male. So there are genes that are sort of located on these sex chromosomes, and so we refer to them as sex-linked genes. And the X chromosome, for instance, carries a whole slew of genes that are attached to that X chromosome that control a whole bunch of traits that are completely unrelated to sex. So for instance, in fruit flies, if you have a fruit fly that has a white eye color, this is a recessive trait that is inherited from the X chromosome. Now, eye color doesn't have anything to do with sex, but it is a sex-linked gene because the gene for eye color here is attached to the X chromosome. And so here, if we look at that sort of process, here you have a female. She's got red eyes. She's got that kind of red gene linked to her two X's. Here you have the male. He has the white-eyed allele on his one little X chromosome. And so sort of depending on what results, you know, if we get the red dominant and the white recessive on those two X's, then the resultant offspring will have red eyes. If you have a male offspring, since the Y doesn't have the sort of white eye color gene on it, all they'll get is the red gene on the X chromosome, and so the resultant offspring, both male and female, will be having red eyes. However, if we then, that's in our F1 generation, if we look then at our F2 generation, and we take this female who is carrying that recessive white eye color gene attached to one of her X chromosomes, if she then mates with another male, there is a 25% chance that she will pass on her X chromosome with the white recessive eye color allele to a male who is then only going to have a Y chromosome and not going to have the eye color, in which case the eye color for this particular offspring would be determined by the X chromosome, and they'd have a 25% chance of having an offspring with white eyes. And then if you had a male who had that X chromosome with the white eye allele, then he would have a 50% chance of passing on that allele and have it actually show up in the eyeball, because it is, of course, a recessive trait, not hidden by the red dominant color. Most of our sort of sex-linked disorders in humans tend to be affecting primarily males, right? A lot of these are recessive traits, so they don't tend to show up unless you have a recessive allele on that X chromosome, and since men only have the one X chromosome, if they get the X chromosome with that recessive allele, their little Y chromosome sort of can't, dominate, and so they get that X-linked recessive allele from their mother, and so they end up with the disorder because the Y chromosome doesn't have the allele at all. Conversely, females, because a lot of these traits are recessive, they have to get the allele from both the X chromosome that they get from their mother and the X chromosome that they get from their father. So hemophilia sort of happens to be one of these traits. So you can see this happens to be in Queen Victoria in Victorian England. She was married to Albert. They had a couple children. Alice was a carrier, just like the Queen Victoria was a carrier for the hemophilia. Louis was not. He did not get the sort of sex-linked gene. He was normal. I'm doing this wrong, guys. I'm having a brain fart. Let me try this again. Sometimes my brain just does not function properly. Queen Victoria was a carrier for the hemophilia allele, but because she had one sort of X chromosome with it, one X chromosome without it, she was normal. She had children with Albert. One of those children was Alice. Alice was also a carrier because she got the hemophilia allele from her mother. When she had children with Louis, one of those children was Alexandra, who was also a carrier. You see the women are kind of carrying it. She then, Alexandra, had children with Nicholas II of Russia, and their child, Alexis, male, got the recessive allele from his mother on his X chromosome that he got from his mother Alexandra. And since he didn't get an X chromosome but a Y chromosome from his father, the recessive allele is able then to show up. So that's how a recessive X-linked trait works, such as hemophilia. There are some traits that are sort of dominant traits that can be passed on via sex-linked genes, but mostly they tend to be sort of recessive X-linked disorders. Conversely, if we look at our male chromosomes, our Y chromosomes, we can actually look at evolutionary history, because essentially the Y chromosome is passed on intact from father to son to son to son to son, et cetera, et cetera. And so they did this study back in 2003 where they collected DNA from a whole slew of people all over Central Asia, and they discovered about 8% of the people in this part of the world have a very similar Y chromosome. They did sort of some more analysis, kind of following the genes back through time, and they ended up on a man who lived about 1,000 years ago. They then looked at the historical records of who was possibly living at that time, and speculated that the ruler Genghis Khan, who was a Mongolian kind of ruler back in the Far Ages, had passed on his Y chromosome to his descendants, who then passed on that Y chromosome to their descendants, who then passed it on to their sons and to their sons and to their sons, and to nearly, in the end, 16 million male descendants, ultimately from what their best guess is from Genghis Khan, which is kind of just astonishing when you think about that. So to sort of wrap up this chapter, we started off with sort of that blending hypothesis and pangenes and why that doesn't work. We talked about terminology in terms of our P, F1, F2 generations, hybrids versus kind of true breeds, right? True breeds meaning the homozygous dominant and the homozygous recessive, and then the hybrids being that heterozygous. We also talked about homozygous versus heterozygous, dominant versus recessive, genotype and phenotype. You should definitely be able to do a Punnett square from a monohybrid cross. To be able to explain the law of segregation, the genetic relationship between the homologous chromosomes, how Mendels' law of independent assortment applies to those dihybrid cross situations when we're looking at two traits, sort of the probability of an event when we do a Punnett square, how we use sort of pedigrees and family trees to look at human traits, recessive and dominant disorders. We talked a little bit about fetal testing. Then those sort of oddballs, right? Incomplete dominance, multiple alleles, codominance, pleiotropy, polygenic inheritance, sort of how it, runs sort of counter to Mendel. The sickle cell and its adaptivity, it confers sort of a benefit in terms of being a carrier of the sickle cell allele to surviving malaria. How chromosomes are sort of behind our theory of inheritance. At the end here, we talked about sex-linked genes. And we talked about some sex-linked disorders, and then ended up talking about the Y chromosome being used to sort of trace our human ancestry. And that sort of wraps up chapter 9.